I really like this picture here. So this is actually a 5-year work of Phantom 5, actually led by Alistair Forrest, in a single UMAP slide. So here, every single dot is a sample that was generated in profile using cage technology. And you can actually annotate individual dots here to understand the biology. So you can see that mesenchyme cells cluster together and they differentiate into adipocytes, endothelium, smooth muscle cells, and so on. On the right-hand side, you have the epithelium. There's clustering with various cancer-related carcinomas. And on the bottom, you have the whole cluster of hematopoietic system. So Phantom 5 was pretty comprehensive, covering around 400 human cell types, profiling in bulk. Of course, now we are here at the Human Cell Atlas meeting. Can we expand this map at the single cell resolution and be able to profile as more cells and discover new cell types? So to achieve this, we're still very much interested in looking at gene expression profiles at the 5' end, mainly developing this cage technology. So we have published this work in Fluidigm-based, and now effectively you're able to do this 5' strategy also in 10x droplet system. And I'm going to explain to you what kind of data you get from this method. So here is a classical expression of MYC loci in the genome browser. In the first row, you have the 3' base, where you have very nice enrichment at the tail of the MYC. But in the bottom 3 rows are different. First 2 are the 10x. We use poly-DT random reverse transcription, as well as the hexamer random primary reverse transcription. And you can see that very nice sharp TSS peaks that recapitulates the bulk cage strategy. So we want to kind of expand this for 2 very reasons. One is to understand where these promoters are, because once you then understand where these transcription vectors bind to these promoters, that will then activate gene transcription and be able to understand gene regulation. But we have also shown that these transcription factors can also bind to upstream or downstream of these promoters to then elicit bidirectional gene transcription called enhancer RNAs. So effectively, there are transcription factors bind to enhancer regions and starts transcribing RNA, both positive and negative strands. So we have shown this using single cell cage technology, and you can observe and appreciate this bimodal distribution of plus and minus enhancer RNAs in bulk and as well at the single cell cage level. And what's interesting is that you can now break this down into single cell resolution, and you can see that individual cell either has one strand, positive strand, or the negative strand. You rarely see a single cell expressing both positive and negative strand, and you can actually validate and visualize this by labeling single molecule RNA fish and single molecule resolution of enhancer RNAs, and they recapitulate very well to the single cell cage gene expression analysis. So such method could also be, again, observed using 10x droplet-based. Here you have the fibroblast and IPS. Both poly-DT and random priming, surprisingly, can reveal both enhancer RNA profiles. And surprisingly, again, that the number of enhancer RNAs that you detect are rather similar, surprisingly, because you would expect a lot of these enhancer RNAs are poly-A minus. But even using poly-DT priming, we are able to detect these enhancer RNAs, potentially suggesting that some of these enhancers are more stable than we previously thought. So we decided to take this technology to profiling a little bit more complex tissues, PBMCs, and we stimulated with a PMA. Basically, we activated the immune response in PBMCs, and we performed 5' single-cell RNA sequencing. So here you can see different populations, both in control and stimulation, and you can actually quantify this. And you can see an increase in number of B cells and CD8 T cells, but also decrease in CD14 monocytes, as you would expect, because PMA actually differentiates monocytes to macrophages, and that CD14 population is decreased. But we are more interested in gene expression changes and what are the promoters that are activated. And here, NR4A2 is one of the main transcription factors that activates, that differentiates Tregs, basically. And you can see there's a strong expression of NR4A2 upon the stimulation of PMA compared to the control. Here on the bottom right is the promoter expression. And surprisingly, if you look at the upstream of NR4A2, you also can quantify and detect these enhancer RNAs, and we can visualize this in the genome browser. So for instance, here is the NF4A2 locus. The transcription is going from right to left. And you can see this purple transcriptional start sites at the single cell. And this is the average single cell expression. The next track here you see is the epigenome roadmap DNA's hypersensitive site. So basically every red region here represents open chromatin. So you can see a nice overlap with the promoters with the open chromatin, where if you look at upstream, you do expect an open chromatin there. And surprisingly, if you zoom in and scale into these regions, we are able to identify these bimodal, bidirectional enhancer RNAs nicely overlapping to one of the open chromatin regions, suggesting those are potentially functional enhancers for this particular cell type. Now that we have this in single cells, we can stratify across multiple cell types of PBMCs, and you can appreciate that these promoter enhancer pairs are very cell type specific here in NK cells or T cells, and moderately in monocytes. What we could also appreciate is you can expand this genome browser. Here is the same NF4A2 locus, and if you look at the upstream of it, there's another open chromatin region there. So what you can really appreciate is that even in the control and the stimulated, there is an enhancer RNA activities, but absolutely absent in the control setting, but only in the stimulated. You gain this enhancer promoter interactions upon stimulation. So it suggests to show that these 5' strategies allow us to not only detect gene expression profiles, but even looking at enhancer activities in a dynamic fashion. So if you're performing single-cell TAC-seq as well as single-cell RNA-seq, you may have to prepare 2 separate libraries. I like to think that you can kill 2 birds with 1 stone by just profiling 5' single-cell RNA sequencing and study the promoter-enhancer relationship there. So it's kind of a very hypothesized model here, where on stimulated fashion you have an innate, already a minor interaction with the promoter and an enhancer that is driving the NF4A2 at the basal level, but upon stimulation, there's definitely an activation of new enhancer regions that synergizes the contact points that could potentially increase the expression of NF4A2. So this is quite of an interesting understanding to see how these enhancers could be gained or lost in a dynamic fashion, and hopefully a single cell 5' strategy can capture this. We have another example of doing this in brain organoids. So we take healthy human iPS cells, we differentiate them into 3D organoids, and you can appreciate these complex structures even day 18. If you stain them with MAP2 mature neurons or PEC6 progenitor cells, there are different compartments within these organoids. And you can also perform 5' strategies to identify transcription factors that are specific to a cell type and their nearby enhancers. What's surprising is that a lot of these are also human gain enhancers. So these are enhancers that are not even expressed in primates. So you're able to understand potential gene regulation in human psychological SNPs that are usually found in human gain enhancers. So what I've asked a student of mine and also the postdoc who have helped this to help me analyze this and visualize this in a more comprehensive manner. So I keep asking him, can you do this and that? And he got frustrated at me, and basically he wrote a Shiny app. Basically you can just upload the data and select any clusters you want, and it will make a pseudotime for you. And so it's really based on Monaco 3, and kind of going back to earlier discussion with Jonah, there are already a lot of algorithms out there, but the usability and exploration is still challenging. So what this platform does is that it's a user exploratory tool, so you can kind of navigate and select the clusters that you want and press go, and it will just create a pseudotime. So here's one example. We can select any clusters that we want, and then click go, and it will create this trajectory. You can select different type of clusters and click go, and it will create a different trajectory. So it's really for scientists or biologists who want to understand single cell data. I think this usability and exploratory option is very critical. So you can do this, identify the enhancers and promoters of that, here in NeurG2 are expressed along the pseudotime and the DCX1, whereas the CCND1, which is cell cycle gene, are lost during the differentiation of the neurons. You can also identify modules that are having similar gene expression patterns, and these modules then can become very important when we look at promoter enhancer activities. So here's an example of one transcription factor ZNF and its nearby enhancers, and they have a very similar trajectories in the same module. So you can overlay these and understand if there's any wave of promoter enhancer activities, which is very analogous to one of the Phantom papers where we looked at how enhancers are activated just before the promoters. And can we use then single cell RNA sequencing 5' strategy to understand the dynamics of enhancer and promoter interactions. So hopefully this user Shiny app will be available to many of you. Again, this is just examples of what you can select from this. And hopefully this will make a lot of biologists happy to engage and explore the data on their own. So it's my summary slide. Hopefully I could convince you that 5' approach could really reveal both promoter and enhancers, and that we can observe this additive enhancer engagement upon stimulation of immune cells. Hopefully this exploratory UI hopefully will be expanded more through different algorithms that are out there, and that it's very interesting and further study needs to be done, but how we can understand promoter enhancer dynamics and how phasing effects can regulate gene expression will be of great importance. So with that, I want to thank again Kono, Joachim, and Yoon, who have done a lot of work. And I want to thank also Jonathan Moody is also here, and Chung Cha Hong, who've done also a lot of the bioinformatics work on PBMCs. I also want to thank Ando Yoshinari, who's here, who have just doing a lot of coordination and support for the human cell atlas in Japan. So with that, thank you for your attention.